Every plant, animal, and fungus on Earth, including you, is built from cells packed with tiny power stations called mitochondria, and in plants, solar panels called chloroplasts. But here's the twist scientists have proven. These organelles didn't originally belong to your cells at all. They were once completely separate, free-living microbes. This is the endosymbiotic theory, one of the most mind-bending ideas in evolutionary biology, and today we're breaking it down step by step. We'll explore how a simple, primitive cell captured other organisms and, instead of digesting them, formed a permanent partnership that changed the trajectory of life on Earth forever. By the end of this video, you'll understand exactly why scientists say we are literally made of microbes. Let's unpack the origin. Life first appeared on Earth roughly 4 billion years ago, but for a long time, it stayed simple. Just basic prokaryotic cells with no nucleus and no internal organelles. So how did we get from that simplicity to the complex eukaryotic cells that make up your body today? The endosymbiotic theory proposes a radical answer. Instead of slowly evolving complex energy machinery from scratch, early cells took a shortcut. They engulfed existing free-living microbes and, rather than destroying them, formed a permanent symbiotic relationship. Over time, some of the defining organelles inside today's complex cells, mitochondria and chloroplasts, trace their origin directly back to these once independent prokaryotic microbes. Before mitochondria or chloroplasts ever entered the picture, there had to be a host cell capable of engulfing other organisms. Scientists believe this first eukaryotic host resembled a primitive amoeba, a flexible, shape-shifting cell capable of phagocytosis, meaning it could physically engulf prey by wrapping its membrane around it. This same flexible outer membrane began folding inward, increasing the cell's internal surface area dramatically. At some point, a piece of this enfolded membrane pinched off completely, sealing away the cell's chromosomes inside a protective double membrane. This event marks the formation of the very first nucleus, the defining feature that separates eukaryotic cells from simpler prokaryotes. With a nucleus in place, this amoeba-like host continued doing what it did best, engulfing smaller organisms for nutrients. At some point, it ingested a particular type of bacteria, one capable of aerobic respiration, meaning it could efficiently generate energy using oxygen. Normally, engulfed prey would simply be digested and broken down. But this time was different. Instead of being destroyed, the ingested bacteria survived inside the host. As Earth's atmosphere became increasingly oxygen-rich, these aerobic bacteria turned out to be an enormous advantage, giving the host cell a dramatically more efficient way to produce energy. Over countless generations, this relationship deepened into permanent dependency. The story doesn't end with mitochondria. Some early eukaryotic cells, already equipped with mitochondria, went on to acquire a second endosymbiont, photosynthetic bacteria capable of converting sunlight directly into chemical energy. Just like the aerobic bacteria before them, these photosynthetic microbes weren't digested. They survived within their host, forming another lasting symbiotic relationship. Over evolutionary time, this partnership gave rise to modern chloroplasts, the organelles responsible for photosynthesis in plants and algae today. Every green leaf you see is powered by the living descendants of ancient bacteria captured and repurposed billions of years ago. Becoming a permanent organelle came at a cost. Over billions of years of assimilation, these once fully independent bacteria gradually lost features they no longer needed inside their protected host environment. They shed their rigid cell walls and lost the vast majority of their original genetic material, since genes for independent survival were no longer necessary when the host cell provided protection and resources. What started as a fully equipped, free-living ancient bacterium slowly transformed into a streamlined organelle with only a small, faint remnant of its original circular DNA. The trade-off was absolute dependency. Today, mitochondria and chloroplasts cannot grow, replicate, or survive outside their host cell. If mitochondria really were engulfed after the nucleus had already formed, evolutionary theory predicts we should be able to find primitive, living cells that capture this exact transitional phase cells with a nucleus, but no mitochondria yet, and remarkably, we have. Several primitive eukaryotic microbes, including Giardia and Trichomonas, possess a true, fully formed nuclear membrane, yet completely lack mitochondria. These organisms represent what scientists call the mitochondriate stage of cellular evolution, a snapshot of what early eukaryotic cells may have looked like before they acquired their aerobic bacterial partners. Finding real living examples of this predicted transitional stage is powerful, supporting evidence for the sequence of events proposed by the endosymbiotic theory. One of the most compelling pieces of evidence for the endosymbiotic theory is remarkably simple: size. The physical dimensions of mitochondria and chloroplasts don't match the eukaryotic host cells they live inside. They match the proposed ancient bacterial ancestors instead. Both mitochondria and chloroplasts measure roughly 1 to 10 micrometers, which is precisely the same size range as modern free-living prokaryotic cells. This is a striking 1-to-1 scale match. If these organelles had evolved from scratch inside the host cell, there'd be no particular reason for them to match bacterial dimensions so closely. Instead, this consistent sizing strongly suggests that mitochondria and chloroplasts are, quite literally, shrunken and streamlined versions of the free-living bacteria. Here's a detail that's hard to explain unless the endosymbiotic theory is true. Mitochondria and chloroplasts don't replicate the way the rest of the host cell does. Instead, they replicate exactly like bacteria, dividing independently through binary fission rather than syncing with the host cell's own division cycle. Even more strikingly, chloroplasts and the mitochondria of certain protists use matching FT's proteins or close FT's homologs positioned right at their division plane, the exact same molecular machinery free-living bacteria use to split themselves in two. This isn't a coincidence. It's a genetic fingerprint left over from these organelles' bacterial past. Perhaps the strongest evidence of all comes from genetics itself. When scientists compare ancient prokaryotes, modern organelles, and the eukaryotic host cell side by side, a clear pattern emerges. Ancient bacteria have circular DNA, and so do mitochondria and chloroplasts, which carry their own distinct circular DNA completely separate from the host genome, which is linear. The pattern repeats with ribosomes. Bacteria use 30S and 50S ribosomal subunits, and remarkably, mitochondria and chloroplasts use their own distinct 30S and 50S ribosomes too, while the eukaryotic host cell itself uses entirely different 40S and 60S subunits. Genetically and structurally, organelles are undeniable matches to ancient bacteria. While the bacterial origins of mitochondria and chloroplasts are now well established, some scientists believe endosymbiosis might extend even further. One hypothesis suggests modern cellular mobility structures, flagella and cilia, the whip-like tails and hair-like structures cells use to move, may have originally come from engulfed spirochetes bacteria, known for their distinctive corkscrew shape and movement. It's a compelling idea, extending the endosymbiotic story into the architecture of the modern cell. So what does this all add up to? The endosymbiotic theory proves something extraordinary. The evolutionary leap to complex life was achieved not through isolation, but through assimilation. Mitochondria and chloroplasts, found inside today's eukaryotic cells, were once entirely separate, free-living prokaryotic microbes. By trading away their independence for the safety and resources of a host cell, they became the biological engines that now power every modern plant, animal, and fungus on the planet, including you. Every breath you take is powered by mitochondria, descended from ancient bacteria.